Hello, hi, I'm Sandhya. I'm your botany lecturer. In this video, we are going to study about the nucleus. Nucleus is somewhat like it's a brain of the nucleus. It's a brain of the cell actually. It's a brain of the cell. It is the most important part of the cell. The vital functions of the cell are controlled and regulated by the nucleus, because this nucleus is having the chromatin reticulum genetic material which is going to control all the activities of the cell. So it's somewhat like a brain of the cell and matter. This nucleus was first discovered by Robert Brown. It was first discovered by Robert Brown, and this nucleus is going to have the chromatin reticulum. That chromatin was first stained by the basic dyes by Fleming. Chromatin reticulum was first stained by the nucleus is first stained by the basic dyes because of the presence because of the presence of the chromatin. Okay, so the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. So Fleming was the one who coined the term chromatin, which was stained by basic dyes, and this chromatin is present inside the nucleus. We'll learn about it. Okay, so this nucleus is somewhat a little bit laterally placed, because in plant cell the nucleus is placed laterally because the center of the cell is occupied by the large vacuole in plant cells. So somewhat it is laterally present. Suppose if this is the plant cell, this is the large vacuole. Here the nucleus will be present. Okay, this is about the plant cell. Then this nucleus is covered by a double membrane cell organelle. It is covered by a double membrane. Nuclear membrane. It's covered by a double membrane, nuclear membrane. Just called as nuclear membrane, nucleolemma, or nuclear envelope. Okay, this is the nucleus. This is the outer membrane. This is the inner membrane. In between the outer membrane and inner membrane is nothing but the perinuclear space. The space is called as perinuclear space. The space between the outer membrane and the inner membrane of the nuclear membrane is the perinuclear space. Okay, and what all contents this particular double membrane is enclosing, this particular thing is called as nucleoplasm. Okay, the nucleoplasm or the nuclear contents is enclosed by the double membrane, nuclear membrane. And this nucleoplasm is having a darkly stained body which is called as the nucleolus. Darkly stained body. This body is called as nucleolus. This is nothing but the nucleolus. Then the nucleoplasm is having the chromatin reticulum. There, like this. Reticulum. This is called as chromatin. Like during the interface of the cell, you are going to observe the chromatin in this form, like a network-like structure. Okay, network-like structure. This is chromatin. Okay, so this double membrane nuclear membrane is having small pores. It is having small pores. Okay, these pores are called as nuclear pore, which are useful for the passage of the contents from nucleus to cytoplasm or cytoplasm to the nucleus. And the outer membrane of this particular nuclear membrane is in continuation with the with the endoplasmic reticulum. Yes, we have studied now endoplasmic reticulum like this, which is nothing but the rough endoplasmic reticulum. So rough endoplasmic reticulum is in continuation with the outer nuclear membrane, like this. Okay, so this is rough endoplasmic reticulum, as it is having ribosomes. Okay, then the nuclear pores are the passages through which the movement of RNA and protein molecules are going to take place in both the directions, between the nucleus and the cytoplasm. Then this nucleus, in most of the cells, single nucleus will be present, but in some of them, many number of nucleus will be present. And when many number of nuclei are present in one particular cell, such type of condition is called as xenocytic. Or sometimes two nuclei will be present. That is called as the two nuclei we can observe in the polar nuclei of the embryo sac. In the embryo sac, the central cell is going to have two nuclei, like that. And sometimes many nuclei will be present. So the number will be varying, but most of the cells will be having one nucleus. And in some cells there will be no nucleus at all. They lack nucleus. The cells like erythrocytes of human cells. Erythrocytes of humans, they lack the nucleus. There is no nucleus. So it is called as enucleated cell. It is enucleated cell. And also sieve tubes. Sieve tubes are also enucleated cells. They also lack the nucleus. Sieve tube cells. They also lack the nucleus. Then so this nucleolus is there. No, this nucleus is darkly stained, and it contains mainly the rRNA and proteins are present. It is a site of synthesis of rRNA, and it is non-membrane bound. It is non-membrane bound structure. Nucleolus. Then during the interface of the stage of the cell, there will be a network like chromatin particles are present. Okay, network of nucleoprotein fibers are present, which is called as chromatin network, mesh like, and which in further stages you are going to observe them as chromosomes. They get condensed. First they'll get dissociated and they get condensed to form chromosomes. And these chromosomes will be having the histones. Histones are the proteins of the these are the basic proteins which are present in the chromatin. So chromatin is nothing but it is having DNA plus histones. Okay, these histones are nothing but the proteins. Then in human cell 23 pairs of chromosomes are present. 23 pairs of chromosomes are present, where 23 pairs means 46 chromosomes are present. 46 chromosomes are present. Okay. Then so in these chromosomes, the DNA is 2 meter long thread. 2 meter long thread is condensed in one particular chromosome among its 46 chromosomes. So a single human cell has approximately 2 meter long thread of DNA distributed among its 46 chromosomes. So in 46 chromosomes, 2 meter long DNA is going to be present. That is about the chromosomes. Then based on the so in interface the chromatin is going to be present. Then in other stages like in prophase, like that, this particular chromatin is going to get dissociated, condensed to form chromosomes, and this chromosome is going to have a primary constriction. It is going to have a primary constriction which is called as centromere. This centromere is covered by a disc-like structures called as kinetochores. Like for example, if you take, this is the chromosome, no? So this particular portion is the centromere. It is covered, this centromere is covered by a disc-like structures which are called as kinetochores. So this is centromere. These disc-like structures are called as kinetochores. Okay, so based on the position of the centromere, these chromosomes are divided into 4 types. Okay, so what are those 4 types? Let us learn now. Based on the position of the primary construction of centromere. The first one is metacentric chromosome. The term itself is meta means center, centric. The centromere is in the center. The such type of chromosome is metacentric. That means the centromere is somewhat center to that of the chromosome. Okay, so it can be like this. It can be of V shape also, like this, metacentric. If you take submetacentric chromosome is a chromosome where it is somewhat nearer to the end, and it is somewhat nearer to the end, not at the nearer to the end, somewhat away from the center. So like this. One is shorter, and other one is longer one, like this. Like this. Okay, submetacentric chromosome. Then if you take the acrocentric, we have 2 unequal arms here, equal arms, 2 unequal arms. Acrocentric chromosome is very nearer to that of the end, very nearer to that of the end, like this. Okay, this is acrocentric. This is the centromere. Wait a minute. Like this. Okay, then telocentric means at the end it is present. At the end, at the tip of the chromosome, the centromere is present. Centromere is going to be present. Okay, this is V-shaped now. This is L-shaped. L-shaped, J-shaped. This is I-shaped chromosomes. So based on the position of the chromosomes, these are divided into metacentric, submetacentric, acrocentric, and telocentric. Sometimes what happens, in non-stained structures are present at the tip of the center or at the tip of the chromatid. So this particular non-stained structure is called as satellite. Okay, non-staining secondary construction. It is a non-staining secondary construction which gives appearance of a small fragment, and it is called as satellite. So this is about the types of chromosomes based on the centromere position. Then these chromosomes are going to have the DNA, and these chromosomes appear like a beads on a string. Like the moti harian, the moti string, like beads on a string, they are present. So such type of beads on a string is called as nucleosome. Like for example, like this. So these are the nucleosomes. Okay, these are the nucleosomes, beads on a string. Okay, now we are going to study about the structure, the nucleosome. The nucleosome is nothing but it is going to have DNA and histones are present. Okay, histones are nothing but the proteins are present. Okay, this DNA is double helical structure. It is a double helical structure. Okay, the histones are 4 types of histones are present. Okay, 4 types of histones are present. So, in this nucleosome, what histones are present? H2A, H2B, H3, and H4 are present. Okay, H4 are present. These are 4, no? The copies of them are present such that they form into copies of them are present such that they form into histone octamer. They form into histone octamer. So like this. The copies of them. So now this is a histone octamer. H2A, H2B, H3, H4. So this is the histone octamer now. Histone octamer. 8 are present, okay? 8 are present. And to this histone octamer, the double helical structure, the DNA is coiled twice like this. Like this. Two turns it makes a coil. Otherwise you can draw like this also. Like this also you can draw. This is the histone octamer, okay? To this, and this is the H1, H11, like this. Two turns are there, okay? Two turns are there. Two turns are there in the, this is the H1. This is having the histone octamer. This one is the histone octamer. This coiling part is called as the two turns is called as the, this is nothing but the DNA. It is double helical part, double helical structure, double helix, and two turns are present. In these two turns, 200 base pairs are present. 200 base pairs are present. Okay, 200 base pairs are present, and this is one nucleosome, and to this another nucleosome is going to be attached. Then histone. Like this, another one is going to be another nucleosome is going to be present. So the DNA which is present in between two nucleosomes, this is called as linker DNA. This is called as linker DNA. Okay, linker DNA. And here the DNA is negatively charged, whereas the histones are positively charged. So that they are making a correct packaging and meaningful packaging. Okay, the association between negatively charged DNA and positively charged histones allows the meaningful DNA packaging inside the nucleus. And these beads on string structures in chromatin, they are packaged in such a form to form a chromatin fibers. Then further in next stages of the cell, they are condensed, they are dissociating into chromosomes. This will get dissociated into chromosomes. This is about the nucleosome, chromatin reticulum, and about the nucleus. Now coming on to the microbodies. Microbodies are nothing but peroxisomes and glyoxisomes. These are called as the microbodies. These microbodies are present in most of the plant cells. They are spherical structures. They are membrane bound spherical structures. Then these peroxisomes are useful for first peroxisomes we are going to study about it. About the peroxisomes we are going to study. These are useful for the catabolism of the long chain fatty acids. Hence they are useful in the synthesis of phospholipids. So they are useful in the synthesis of phospholipids, and also they are useful for and also they are going to involve in the photorespiration and protection of cell. They are going to involve in photorespiration and protection of the cell. These are the functions of peroxisomes. Peroxisomes are the spherical structures, membrane-bound structures, and they are helpful in the catabolism of the long-chain fatty acids. They are useful in the synthesis of phospholipids, and they are involved in the photorespiration and protection of cells from toxic effects of hydrogen peroxide. Toxic effects of hydrogen peroxide. Then coming on to glyoxisomes. Glyoxisomes are generally present in rich germinating seeds, fat-rich germinating seeds. One such example is castor. Fat-rich germinating seeds. In castor they are present. They contain the enzymes for glyoxylate cycle. They contain the enzymes for glyoxylate cycle, which convert the lipids into. These enzymes convert the lipids into carbohydrates. Okay, so this is about the peroxisomes and glyoxisomes. These peroxisomes and glyoxisomes together form the microbodies. They are commonly found in plant cells. Thank you.